Today I will tell you a story about time, decay, and the eternal question that haunts both kings and poets. Can we live forever? If the thought of immortality stirs something ancient within you, if the idea of defying time's relentless tide feels like a whisper calling through your bones, then stay, like, share, and let this journey begin, where cells whisper secrets and biology bends beneath the weight of dreams. Aging begins not in wrinkles or gray hairs, but in silence, an unseen shift beneath the skin, deep within the crypts of the body's citadels. The human body, marvelous and intricate, is a cathedral built from trillions of living bricks, each a cell pulsing with life, each obeying a choreography written in strands of DNA. When life begins, the dance is fluid, immaculate, a cascade of renewal and repair. But as years fold into each other like brittle parchment, the rhythm falters, the precision loosens, and entropy claims its first quiet victories. From the moment of birth, time etches itself into flesh. Cells divide, replicate, build tissues, heal wounds. But replication is never perfect. DNA, coiled in elegant helices, is copied with exquisite care, yet errors accumulate. Telomeres, the guardians of chromosomes, shorten with each division, like the burnt ends of candles dwindling in the night. They are the biological clocks embedded in our genome, ticking not to mark hours but to count the days we have left before the machinery grinds down. The telomere is a paradox, essential for life, yet a harbinger of death. It protects the genome from fraying, but its erosion limits a cell's life. When telomeres grow too short, cells enter senescence, a state of arrested function. They do not die, but they no longer divide. They linger, secreting molecules that cloud the tissue with inflammation and degradation, contributing to the tapestry of aging. These are the zombie cells, relics of vigor, now haunting the body's quiet corners. But aging is more than cellular senescence. It is mitochondrial decline, a slow decay of the energy furnaces that power every heartbeat and breath. Mitochondria once thrived as independent organisms, ancient symbionts swallowed by our ancestors. Now, they burn fuel into motion, but with each passing decade, their fires flicker. Reactive oxygen species, molecular ghosts, are released, damaging lipids, proteins, and DNA. The cells grow weary, the organs tire. Vitality begins to retreat like the last rays of dusk. The immune system, vigilant in youth, becomes muddled with age. Its memory clouds, its response dulled. The orchestra of white blood cells that once surged against infections begins to falter. Old enemies return with new strength. Viruses reawaken. Wounds heal more slowly. Cancers slip through unnoticed. Immunosenescence wraps the body in vulnerability, leaving the fortress exposed. Inflammation, low and constant, settles into the tissues like a fog that never lifts, turning friend into foe, protector into saboteur. Meanwhile, stem cells, once abundant, retreat into quiescence. Their powers of regeneration fade. The bone marrow no longer teems with rebirth. The skin does not rebound as before. The liver's miracles become rare. Even the brain, that shimmering web of thought and memory, begins to dim. Neurons die, synapses weaken, and memories fade like old photographs. It is not just the body that ages. It is identity, the self, the sense of continuity that unravels in strands too fine to grasp. The endocrine system, master of hormonal symphony, stumbles into discord. Growth hormone ebbs, sex hormones wane. Cortisol, the harbinger of stress, lingers too long. The body's chemistry, once a fluent song, becomes a jagged rhythm. Muscles, waste. Fat accumulates in viscera. The skin dries and bruises with a whisper. Bones thin until they shatter under the weight they once bore easily. All these are marks, not of disease, but of time's lawful progression. They are the signatures of biology's unwinding. Yet not all creatures succumb so easily. The naked, mole rat, blind and wrinkled, lives far beyond what its size suggests. It resists cancer, its cells seemingly immune to malignancy. The bowhead whale swims for centuries beneath the Arctic ice, its massive heart beating steadily as decades pass like seasons. Hydra, a tiny freshwater polyp, seems to defy aging altogether, endlessly regenerating, never withering. Nature has written secrets into their flesh, secrets humans now seek to decode. Scientists probe the genome, searching for clues, for mutations that stretch life or delay decline. The FOXO gene family, regulators of longevity, command the cellular response to stress, enabling repair and resilience. The sirtuins, proteins awakened by fasting and hardship, preserve mitochondrial function, stabilize DNA, and silence inflammatory noise. Caloric restriction, a harsh discipline, has shown in model organisms to extend life by altering metabolic pathways, awakening ancient mechanisms that favor survival over excess. In the bloodstream of youth lies another mystery. Experiments in heterochronic parabiosis, conjoining the circulatory systems of young and old animals, reveal a strange alchemy. Old tissues bathed in young blood begin to heal. Muscles regenerate, brains brighten, hearts grow strong. Something within the plasma holds the elixir of vitality. The identity of this fountain is still debated, but it whispers of a future where age might be reversed, where rejuvenation becomes more than myth. Gene editing technologies like CRISPR offer the scalpel to rewrite destiny. By correcting mutations, silencing harmful genes, or amplifying protective ones, scientists dream of sculpting longevity into our biology. But the genome is a delicate manuscript, and editing it brings risks as well as hope. Precision is demanded. One error, one unforeseen cascade, and the cure becomes a curse. Yet the ambition burns bright, to not merely delay death, but to fundamentally reshape the course of human aging. Biotech pioneers venture deeper, crafting senolytics, drugs designed to purge the body of senescent cells. By eliminating these saboteurs, tissues might recover their vitality. Trials in mice have shown promise, lifespans extended, organs renewed. Inflammation recedes, vigor returns. Could a daily pill banish the decay of age, sweeping the body clean of its cellular ghosts? The question is no longer science fiction. It is a matter of dosage, delivery, and time. And still, there is the matter of epigenetics, the subtle architecture of gene expression. Aging is not only a story of mutations, but of changes in how genes are read and silenced. DNA methylation, histone modification, chromatin remodeling, these are the soft codes of time, inscribed not in letters but in punctuation. With age, this epigenetic landscape becomes chaotic. Genes meant to sleep awaken, others fade into silence. But if the epigenome can be reset, the cell might remember youth, return to a more vibrant state. In experiments, cells reprogrammed using Yamanaka factors OSKM, a quartet of transcriptional engineers, have reverted to pluripotency, erasing the marks of age, regaining the capacity to become anything. Applied delicately, these factors might rewind the clock without erasing identity. In mice, early successes have reversed signs of aging in tissues. Eyesight restored, organs revitalized. The promise is profound, yet the path is treacherous. To reprogram is to risk chaos, mutation, or cancer, but to master it is to grasp the keys of time. And still, the brain remains the fortress most difficult to breach. While muscles may regrow and skin may tighten, the mind carries the weight of decades in its folds. Neurodegeneration is subtle and cruel. Plaques, tangles, and misfolded proteins destroy the scaffolds of memory. But research accelerates. Therapies aim to clear amyloid, to restore dopamine, to nourish synapses. Stem cell grafts, brain-computer interfaces, and targeted gene therapies hover on the edge of plausibility. To save the brain is to save the self. As twilight settles across this first horizon, we stand not at the end of life, but at its reimagining. Aging, once accepted as inevitable, is now probed as a malleable process, a consequence of pathways that may be rewired. The question is no longer simply whether we can live forever, but how long we can remain whole, awake and luminous in the face of entropy. For the first time in human history, the idea of bending aging is not myth or poetry. It is becoming biology. Aging, though intricately inscribed into our biology, is not a solitary phenomenon, but a symphony played across countless systems. It is the quiet shifting of molecular harmonies, the creaking of once fluid rhythms. Yet, as science presses deeper into this ancient enigma, the architecture of decay begins to shimmer with the outlines of possibility. The body, long assumed to fall under the spell of time without resistance, now appears to carry within it a code for rebirth, waiting only for the right key. Deep within each cell lies not only DNA, but a landscape of regulators, tiny molecules, and structural features that determine which genes wake and which sleep. These are not mutations in the genetic script, but changes in the way it is read, and this too contributes to the withering of the years. As the epigenetic signature becomes scrambled, so too does the orchestration of cellular identity. A muscle cell forgets its strength. A neuron loses its synaptic fire. A liver cell grows weary of detoxifying the tides of life. But this chaos is not irreversible. The cell retains a memory of its youth buried beneath the scars. Scientists are learning to trace the patterns of methylation and histone modification, using them not only as biomarkers of age, but as switches that can be flipped. Reprogramming factors, when used in moderation, do not erase the cell, but soften the edges of its decline. It is the difference between obliteration and rejuvenation. A careful symphony of expression can, if tuned correctly, breathe vigor back into old tissues. Alongside reprogramming lies the growing discipline of systems biology, which does not view aging as one mechanism gone awry, but as a collapse of equilibrium across the body's networks. Organs speak to one another through hormones, neurotransmitters, cytokines, and peptides. When these messages become distorted, the body no longer maintains coherence. Homeostasis unravels. To restore youth, then, is not to fix a single part, but to rebalance the whole, to rewrite the logic of the body's internal conversation. At the center of this systemic vision stands the metabolome, the sum of every chemical reaction occurring within us. The flux of glucose, the ebb of insulin, the swell of amino acids, the flow of lipids, all shape how cells function and survive. With age, metabolism becomes less flexible. Cells lose their ability to shift between energy sources. Inflammation stiffens the body's response to stress. The exquisite dance of anabolism and catabolism begins to drag. But metabolic flexibility, once lost, may be rekindled. Intermittent fasting, ketogenic diets, and time-restricted feeding. Regimes aim not just at weight control, but at resetting the cellular rhythm. Fasting mimics the evolutionary pressures of scarcity, awakening genes that protect and repair. Autophagy, the process by which cells digest and recycle their damaged components, is induced not by excess, but by restraint. It is the body's way of cleansing its own debris. As this self-devouring mechanism awakens, cells regain clarity. Misfolded proteins vanish. Mitochondria are refreshed. The mitochondrion, long known as the powerhouse of the cell, is more than an energy factory. It is a decision maker, sensing cellular stress and choosing between survival and death. When mitochondria falter, energy declines, and the signals they emit turn chaotic. But emerging research explores how these organelles might be regenerated, either through biogenesis or through transplantation. Mitochondrial replacement, though still experimental, hints at the possibility of revitalizing tissues from their energetic core. And here, the conversation turns toward the very mechanics of death. Apoptosis, the orchestrated suicide of cells, is essential in youth, pruning the weak, sculpting the body, eliminating threats. But with age, this balance is lost. Too much cell death, and tissues fail. Too little, and dysfunctional cells accumulate. Therapies are being developed to recalibrate this balance, to ensure that cells die when they should, but also survive when needed. The goal is not mere survival, but resilience, a capacity to respond with elegance to stress and injury. In the bloodstream, beyond cells and hormones, flow microvesicles, exosomes, and RNA fragments, tiny messages that traffic between organs, delivering instructions and reflections. These molecular couriers, once overlooked, are now seen as critical in the aging dialogue. They carry the voice of youth or the warning of decline. Scientists are mapping this cellular language, hoping to engineer messages of renewal. Infusions of youthful exosomes have begun to show promise in animal studies, suggesting a future where blood itself becomes medicine. The field of geroscience has risen in response to these revelations, not seeking to cure individual diseases, but to confront their shared root, aging. By targeting the fundamental pathways of senescence, scientists hope to delay the onset of multiple diseases simultaneously: heart failure, Alzheimer's, diabetes, arthritis, not with separate drugs for each, but with interventions that maintain the integrity of life's foundation. It is an audacious vision, one that redefines the goal of medicine. Yet challenges remain. For while the body is mechanical in some respects, it is also adaptive, evolutionary, and poetic. Aging may serve purposes we do not fully understand. It prunes populations, preserves genetic diversity, and may even play roles in species-level evolution. To defy aging is to intervene not only in biology, but in the trajectory of human history. If death recedes, what becomes of meaning, of urgency, of the arc that bends a life toward legacy? Still, the pursuit advances. Venture capital flows into longevity startups. Clinics offer gene therapies. Wearable devices track the molecular shadows of time. Artificial intelligence combs through genomic data to find patterns, predict lifespan, and customize anti-aging protocols. The dream of personalized immortality, tailored supplements, gene editing, cellular rejuvenation is no longer relegated to myth. It is in part a market, but also a mirror reflecting our deepest fears and desires. At the philosophical core lies the question of identity. If the body is continuously renewed, is the self preserved? Are we the continuity of our memories, or the vessel that carries them? If neurons are replaced, if brain tissue is regenerated, does the soul remain intact? These are not trivial puzzles, but ones that science may be forced to answer. Rebuilding flesh is one thing, preserving consciousness is another. The biology of aging does not exist in isolation from the mystery of awareness. The future imagined by longevity science is one of radical choice. Not only longer lives, but healthier ones. Centenarians who dance, elders who create, minds that remain sharp for centuries. Yet this vision must be tempered with ethics. Who receives these treatments? Who pays? How does society adapt to lives that stretch into the hundreds of years? Will age become privilege? Will youth become obsolete? The pursuit of eternal life must wrestle with justice as much as with biology. Nature, in its wisdom, has made aging inevitable, but it may not be immutable. The winds are shifting. Where, once we measured life in decades, we may soon think in centuries. Where once death stood as the final curtain, we may begin to see it as a negotiable horizon. The first human to live beyond 150 years may already walk among us, or they may be born tomorrow, with their destiny altered by choices made in laboratories today. To live forever is not to defy nature, but to collaborate with it, to understand the threads it weaves, and to guide them with grace. Longevity is not about escaping death, but about deepening life, making each moment more vivid, each breath more resilient, each heartbeat echoing a longer song. In the biology of aging lies a new frontier, not just of science, but of humanity's place in time. Beneath the surface of aging lies a hidden architecture of decay and renewal, a latticework of cellular crosstalk and biochemical loops that has shaped the arc of every human life since the dawn of time. But now, under the piercing light of modern biology, the scaffolding that holds the illusion of inevitability begins to fracture. The veil lifts, and behind it, The intricate mechanics of aging reveal themselves as both map and maze, a code that can be studied, deciphered, and perhaps rewritten. At the core of this unfolding story lies the concept of cellular senescence. When cells are damaged, or when they have divided too many times, they enter a state of arrest, no longer dividing, no longer functioning yet stubbornly alive. These senescent cells become biochemical saboteurs, secreting inflammatory signals, enzymes, and growth factors that erode surrounding tissue. They are not merely relics of time, but active participants in the body's breakdown, whispering ruin to neighboring cells, inciting a slow cascade of dysfunction. But these senescent cells can be identified. They wear molecular signatures, P16, beta-galactosidase, and other markers that make them distinct. And more importantly, they can be eliminated. The rise of senolytic therapies aims to clear the body of these zombie-like remnants, restoring tissue vitality and staving off the diseases of age. In mice, the removal of senescent cells has led to improved cardiac function, enhanced physical endurance, even longer lifespans. What once seemed irreversible, the slow rot of age, now appears to be, at least in part, reversible. Yet even as old cells falter, new ones can be summoned. The body retains pools of stem cells, ancient, undifferentiated, regenerative, scattered through bone marrow, muscle, fat, and brain. These are the architects of healing, ready to divide and specialize when the body is wounded. But with age, stem cells become fewer, more sluggish, less willing to awaken. They reside in niches that wither over time, their environments poisoned by inflammation, fibrosis, or metabolic stress. Still, the dream of stem cell therapy endures. Scientists are now learning how to harvest these cells, expand them in culture, and reintroduce them into damaged tissues. Pluripotent stem cells, which can become any cell type in the body, have been created from adult cells, opening the door to personalized regenerative medicine. It is a delicate art, rebuilding without mutation, encouraging repair without triggering malignancy. But the direction is clear. Aging need not be a static fall. With the right cues, the body may be coaxed to rebuild itself from within. Beyond, stem cells, another silent architect, shapes our fate, the immune system. It is our shield, our memory, our sentinel, and it too ages. The phenomenon is called immunosenescence, a gradual weakening of immune vigilance. With age, T cells lose their ability to respond to new threats. B cells forget how to craft potent antibodies. The innate immune system becomes overzealous, firing inflammatory bullets even in the absence of danger. This imbalance not only increases vulnerability to infections, but fuels chronic inflammation, what scientists call inflammaging. Chronic inflammation is a low-grade fire that never extinguishes. It damages blood vessels, corrodes cartilage, disrupts insulin signaling, and accelerates neurodegeneration. But it also offers a target. Anti-inflammatory compounds, some natural and others pharmaceutical, are being studied not merely for symptom relief, but as foundational anti-aging strategies. Curcumin, metformin, rapamycin. These molecules act like whispers to the immune system, telling it to ease its grip, to soften its tone, to preserve rather than destroy. Metformin, in particular, has emerged as a symbol of this new approach to aging. An old diabetes drug, cheap and well tolerated, it seems to do more than regulate blood sugar. It modulates energy sensors in the cell, mimicking the effects of caloric restriction, reducing oxidative stress and dampening inflammation. In population studies, metformin users live longer than their non-diabetic peers. A clinical trial, called TAME, targeting aging with metformin, is underway, not to treat disease but to treat aging itself, marking a bold step into uncharted territory. Then there is rapamycin, a compound first discovered in the soil of Easter Island. It inhibits mTOR, a master regulator of growth and metabolism. Inhibiting mTOR slows cellular growth, activates autophagy, and extends lifespan in a host of organisms. From yeast to mammals, rapamycin does not make cells stronger. It teaches them restraint. It tells them to clean house before building anew. And in that cleaning, in that internal pause, cells find resilience. These compounds suggest that aging is not a singular event, but a system in tension, a network of switches and balances that can be nudged. A little inhibition here, a little activation there, and the system begins to sway differently. It is not immortality that we chase, but modulation, a gentle tuning of time's instruments so that they play longer, clearer, and with fewer discordant notes. Alongside these interventions grows the field of genetic engineering. CRISPR, the revolutionary tool that allows precise editing of the genome, holds immense promise for aging. Mutations associated with progeria, a rare premature aging syndrome, have already been corrected in cells. More broadly, scientists are exploring how to enhance protective genes, like those in the FOXO family, and silence harmful ones, such as those promoting inflammation or instability in DNA repair. And yet, even with the power to edit our blueprint, the question remains: which edits are wise? Evolution has sculpted us over millennia, trading longevity for fertility, adaptability for fragility. To re-engineer the genome is to dance with forces that lie deep beneath our conscious design. Each change may have unforeseen consequences. Still, the potential is vast. If the body is a script, then perhaps it can be revised, extended, its final chapters made more graceful. But the genome is only the beginning. Just as important is the proteome, the collection of proteins that do the actual work of life. Proteins fold, interact, degrade, and regenerate. With age, misfolded proteins accumulate, clumping into plaques in the brain, tangles in muscles, dysfunction in the skin and eyes. The body's systems for clearing these errors, chaperone proteins, proteasomes, autophagy pathways, begin to falter. Restoring protein homeostasis becomes essential. Enter the realm of heat shock proteins, molecular guardians that help other proteins fold correctly and dispose of those beyond repair. These guardians can be induced by stress, heat, fasting, exercise, even certain supplements. The irony of biology is that mild stress often yields strength. Hormesis, this principle is called. The idea that challenge builds resilience. A touch of poison becomes a cure. A flicker of damage awakens the machinery of defense. So too does sleep. It is not idleness, but symphony. A time when the brain clears debris, when hormones recalibrate, when the heart and lungs fall into synchrony. Chronic sleep deprivation accelerates aging, while deep, consistent rest preserves mental acuity and emotional regulation. Sleep is not an accessory to life, but its silent protector. In the quest to live longer, sleep must be revered, not sacrificed. The story of aging is not linear, not a descent from youth to dust. It is a dance of systems, of loss and compensation, of breakdown and repair. And now, as science begins to understand these systems not as immutable, but as modifiable, the narrative begins to shift. The aging body is not a tomb, it is a dialogue, and that dialogue can be rewritten. The question, then, is not simply can we live forever, but how shall we live longer? With what kind of vitality? With what kind of clarity? With what kind of purpose? For it is not only the span of years that matters, but the quality of their unfolding. The path ahead is illuminated not by fantasy, but by evidence. A future of longer, healthier lives is not a myth, it is a horizon. In the expanding inquiry of how to extend the vitality of human life, a quiet revolution is taking place deep inside the machinery of the cell, within the thread-like power plants known as mitochondria. These ancient organelles, once free-living, organisms have become indispensable allies, converting the food we eat into energy, fueling every heartbeat, every breath, every thought. Yet with age, mitochondria falter. Their DNA mutates, their efficiency wanes, their byproducts, reactive, oxygen species, accumulate and scorch the delicate interior of the cell. This mitochondrial decline is not a side effect of aging. It is its harbinger, its accelerant, its silent engine. Mitochondrial dysfunction is the kindling for many fires: neurodegeneration, metabolic diseases, cardiovascular failure, and cancer. Cells starved of energy falter in their duties. Tissues become inflamed, the immune system destabilized. And so, to understand the aging process, one must walk the winding labyrinth of mitochondrial health. One must peer into the bioenergetic core of life and seek ways to rekindle its fading flame. Emerging therapies now attempt to restore this mitochondrial harmony. Some target the organelles directly, introducing compounds that act as antioxidants, designed specifically to reach mitochondrial membranes. Others coax the cell to produce more mitochondria through biogenesis, stimulating the production of new energy factories by activating genetic switches such as PGC1A. Exercise, fasting, and cold exposure have long been known to ignite this internal renewal. What was once mere tradition, movement, hunger, endurance, now reveals its molecular justification. Then there is NAD+, a molecule once relegated to obscure textbooks, now heralded as a linchpin in cellular aging. NAD+ serves as a coenzyme in metabolic reactions, but also plays a key role in activating sirtuins, proteins that promote DNA repair, reduce inflammation, and stabilize mitochondria. With E, NAD+ levels plummet, and with that decline comes a cascade: decreased energy production, impaired repair, increased vulnerability to stress. Restoring NAD+ levels through precursors like NMN or NR has become a focal point of rejuvenation research. In animal studies, such supplementation improves muscle strength, cognitive performance, and insulin sensitivity. In humans, trials are ongoing, but the promise is luminous. The sirtuins themselves are emblematic of a new era in biology, one that doesn't fight death blindly, but instead deciphers the code of survival etched into our DNA. These proteins are guardians of the genome, responding to stress, activating repair, and preserving order. They are silent sentinels that whisper across time, guiding the cell through crisis. Caloric restriction, a proven method of lifespan extension across species, activates sirtuins. So do certain plant compounds, such as resveratrol, found in the skin of red grapes. Though much about sirtuin activation remains uncertain, their story continues to unfurl in laboratories around the world. With every experiment, a new stanza in the poem of life's persistence. Yet aging does not merely degrade the energy of life, it muddles the symphony of signaling, the communication between organs, cells, and systems. The endocrine system, the hormonal axis, loses its equilibrium. Growth hormone, testosterone, estrogen, melatonin all decline or misfire. These changes shift the metabolic landscape, increasing fat deposition, reducing bone density, weakening muscle mass, and altering mood and cognition. The aging body becomes a landscape where messages falter and echoes mislead. Hormone replacement. Therapy has long been debated in this context, sometimes praised for restoring vigor, sometimes criticized for its risks. But new approaches seek nuance, not merely flooding the body with hormones, but mimicking youthful rhythms, targeting specific receptors, and tailoring treatments to genetic profiles. Melatonin, in particular, once thought solely as a sleep aid, has been revealed to be a potent antioxidant, an immune modulator, and a regulator of mitochondrial function. Its decline with age may have broader implications than once thought, casting shadows far beyond the circadian rhythm. The circadian system itself, the body's internal clock, is another neglected cornerstone. Every organ follows a rhythm. Every gene oscillates in time. Disruption of these cycles, whether by artificial light, shift work, or chronic stress, accelerates aging. Chronobiology, the study of biological timing, now enters the arena of longevity science. Aligning daily habits with internal rhythms, when to eat, sleep, move, emerges not as lifestyle advice but as cellular medicine. Morning light exposure, time-restricted feeding, and nighttime darkness all serve to reinforce the harmony of the clockwork within. In this recalibration of life's rhythm, the role of the microbiome cannot be ignored. The trillions of bacteria, viruses, fungi, and archaea that live within us form a dynamic ecosystem, influencing immunity, mood, metabolism, and inflammation. With age, the microbiome becomes less diverse, more inflammatory. But just as it degrades, it can be rebuilt. Diet, probiotics, prebiotics, even fecal microbiota transplants are now under study as methods to restore youthful microbial balance. The gut-brain axis, once a metaphor, is now a biological fact, one that weaves cognition and digestion, emotion and immunity, into a single symphony of interaction. Then there are the discoveries that seem to border on the mythic, the partial, reprogramming of cells, the rewinding of the biological clock. Yamanaka factors, 4 transcription factors capable of turning adult cells back into pluripotent stem cells, have now been used to partially reprogram cells in living organisms. In mice, this technique has restored organ function, improved vision, even extended lifespan. The danger, of course, is triggering uncontrolled growth, cancer. But with careful dosing and targeted delivery, the impossible seems less distant. Reprogramming may one day become a way to renew tissues, restore identity, and slow or reverse the signs of time. All these strands, mitochondrial health, sirtuins, NAD+, hormonal balance, circadian alignment, microbiome restoration, genetic editing, cellular reprogramming form not a single path, but a constellation. And in that constellation, a new picture emerges. Aging is not a cliff. It is a garden overrun. And with care, precision, and patience, the weeds may be pulled, the soil renewed, the bloom extended. But to ask whether we can live forever is also to ask whether the systems we inhabit, our healthcare, our economies, our social structures, are prepared for longevity. The body may be willing, but is the world ready? As lifespans stretch, what of health spans? What of purpose, connection, learning, creation? A life of extended years must not become a life of prolonged fragility. The goal is not merely to delay death, but to expand the canvas of life, its clarity, its color, its depth. The burden of aging lies not just in the loss of function, but in the loss of narrative, the shrinking of the world, the fading of memory, the dimming of desire. To live longer must also mean to live deeper, to remain lit from within by curiosity, by love, by art, by mystery. And so, the biology of aging must meet the philosophy of living. It must not sever the soul from the cell, but unite them in a common pursuit. The journey to understand and alter aging is still unfolding. The data accumulate, the tools sharpen, the horizon stretches. What once seemed the province of legend, the elixir of life, the fountain of youth, now becomes the realm of science, not in magic but in molecule, not in dream but in data. And yet the dream persists, reshaped by knowledge, lit by evidence. For the question at the heart of aging is not whether we should live forever, but whether we can learn to age wisely, gracefully, and with intention. Whether we can see time not as an enemy, but as a medium, through which, like artists, we may shape the form of our lives. In the labyrinth of biology, few phenomena are as elusive, as essential, and as endlessly intricate as the process of cellular communication. Beyond the visible fabric of flesh and function, the body is governed by a whispering language of chemical messengers, subtle currents, molecular codes flowing between cells like wind between the reeds. These messages orchestrate everything from tissue repair to immune response, from metabolic rhythm to the delicate dance of growth and decay. But as age advances, these messages fray. The signals grow distorted, or muted, or chaotic. And in this breakdown of conversation, aging plants its quiet seeds. One of the most potent forces in this silent dialogue is inflammation. At its best, inflammation is a healing fire, summoning immune cells to sites of injury or invasion. At its worst, it becomes a smoldering blaze, constant and unresolved, a low-grade storm within the bloodstream. This chronic inflammation, often called inflammaging, is one of the cornerstones of biological aging. It erodes tissues, disrupts metabolism, accelerates degenerative disease. It is not the inflammation we feel, but the one we don't, the kind that simmers below awareness, whispering ruin across decades. Inflammaging arises not from a single spark, but from many. Senescent cells releasing toxic signals, gut microbiota tipping into imbalance, mitochondrial byproducts escaping into circulation, immune cells misfiring, no longer able to distinguish between self and threat. As this systemic inflammation builds, it becomes a driver of almost every chronic condition associated with aging: Alzheimer's disease, type 2 diabetes, atherosclerosis, osteoporosis, even depression. To quell this internal blaze is not a matter of extinguishing, but of rebalancing. Anti-inflammatory interventions, both ancient and new, are stepping forward. Omega-3 fatty acids, long celebrated for heart health, are potent modulators of inflammation. Curcumin, the active compound in turmeric, silences inflammatory genes. Fasting, particularly intermittent and time-restricted eating, reduces circulating inflammatory markers. Exercise lowers inflammation not just through movement, but through complex molecular crosstalk. Even mindfulness and meditation, seemingly separate from biology, reduce inflammation at the genomic level, altering the expression of pro-inflammatory pathways. Alongside inflammation stands another twin force, the immune system itself. With age, it too begins to unravel in a process known as immunosenescence. The youthful immune system is agile, adaptive, and precise. It responds swiftly to infection, clears debris, hunts mutations. But over time it becomes sluggish and confused. The thymus, which educates T cells, withers away. Naive immune cells become rare, while exhausted ones linger. The body loses its ability to mount defenses against new threats while becoming increasingly prone to autoimmunity, attacking itself in a case of mistaken identity. Vaccines become less effective. Wounds take longer to heal. Infections linger. The aging immune system is not just weaker, it is misaligned. To restore it requires more than boosting. It demands re-education. Therapies are emerging that aim to train the immune system anew. From thymic rejuvenation to the infusion of young immune cells, from senolytics that clear the clutter of aged cells to immune checkpoint inhibitors that recalibrate activity. Even the gut microbiome once again reveals its importance, shaping the immune landscape through constant exposure and adaptation. And in the very marrow of this system lies the blood, a fluid of extraordinary complexity. Blood carries nutrients, hormones, immune messengers, and oxygen. But more than a vehicle, it is a messenger itself. In experiments where old and young animals share a circulatory system, a process known as parabiosis, something astonishing occurs. The older animal gains youthful characteristics, improved cognition, increased muscle regeneration, enhanced metabolic health. The younger animal, in contrast, begins to exhibit signs of aging. What travels in blood that could transmit youth? Researchers have found several candidates: GDF11, oxytocin, TIMP2, and others. Molecules whose levels vary with age and whose effects touch multiple organs. Clinical interest now turns toward isolating these factors, replicating their effects, and perhaps developing blood-derived therapies to promote rejuvenation. Not the vampire's myth, but a science of shared vitality. Within the blood also lies another frontier: circulating exosomes and microRNAs, tiny vesicles packed with instructions. These molecular parcels move between cells and organs, altering gene expression at a distance. As we age, the content of these vesicles shifts, promoting decay or renewal depending on the signature they carry. They have become both biomarkers and instruments, tools to read and potentially write the story of aging. At the heart of this dialogue is epigenetics, the modulation of genes without altering DNA itself. The genome is the script, but the epigenome is the director. With age, the epigenetic landscape shifts. Chemical tags like methyl groups accumulate in patterns that promote inflammation, silence repair genes, and unlock dysfunction. This process, called epigenetic drift, is now measurable. Biological age can be estimated more accurately by reading the epigenetic clock than by counting calendar years. And unlike DNA, the epigenome is malleable. It changes with diet, with stress, with sleep, with exercise. Certain compounds like polyphenols, butyrate, or even specific pharmaceuticals can influence epigenetic marks. So can practices like caloric restriction or interval training. The possibility arises not only to measure aging with precision, but to reverse its epigenetic markers, to reorient the trajectory of life toward youth. Beyond molecules and genes lies structure. The physical architecture of the body degrades with time. Cartilage thins, bones lose density, muscles atrophy. But here, too, renewal is possible. Stem cells, once thought to vanish with age, persist in niches throughout the body. They merely fall silent or lose their capacity to respond. Reactivating these cells, whether through signaling molecules, mechanical stress, or reprogramming, becomes central to tissue regeneration. In muscle, satellite cells await cues to awaken. In the brain, neural stem cells can still form new neurons in specific regions. Even in the heart, long believed incapable of renewal, new cells arise, albeit slowly. The scaffold of life is more pliable than once believed. Technologies like 3D bioprinting, bioscaffolds, and organoids suggest a future in which damaged tissue can be replaced not with prosthetics, but with living, growing structures. This shift from repair to regeneration transforms medicine itself from a reactive art to a creative science. But perhaps no regeneration holds more hope than that of the brain. Cognitive decline is among the most feared consequences of aging. Memory fades, reaction slows, attention wavers. Yet the brain is not a fixed machine. It is plastic, adaptable, capable of change. Neurogenesis, once thought impossible after childhood, continues in parts of the adult brain, notably the hippocampus. It slows with age, but it does not vanish. Enriching environments, intellectual challenge, aerobic exercise, and deep social bonds all promote neurogenesis. Compounds like flavonoids, omega-3s, and certain nootropics may further support neural health. Technologies like brain stimulation, transcranial magnetic therapy, and even direct brain-computer interfaces now enter the realm of cognitive enhancement. And as Alzheimer's disease research pivots from plaques to inflammation, metabolism, and synaptic function, new avenues open to preserve and protect the architecture of thought. Cognition is not merely memory, it is identity, presence, imagination. To preserve the mind is to preserve the self. It is not an indulgence, but a necessity, as lifespans stretch and the terrain of aging evolves. For without the clarity of mind, the gift of added years may lose its value. In this ever-widening landscape, aging is no longer a passive decline, but a field of active exploration. From the whispers of genes to the harmonies of hormones, from the scaffolds of muscle to the symphonies of the mind, life's processes reveal themselves not as inevitable decay, but as dynamic systems, capable of resilience, repair, and even renewal. To ask whether we can live forever is to unfold a tapestry of questions about energy, resilience, memory, balance, and growth. Aging is not an enemy, it is a teacher. And to extend life, one must listen, not simply to the metrics of science, but to the quiet wisdom of biology, the architecture of rhythms, the memory of the body, the breath of the cell. Each layer studied brings new tools. Each molecule understood brings new promise. And though immortality remains a distant and perhaps unreachable shore, the voyage toward it redefines what it means to be alive, not merely in years, but in possibility. Beneath the flow of hormones, the beat of the heart, and the flicker of electrical impulses in the brain lies a deeper force shaping the rhythm of life itself: metabolism. A fire at the core of every cell, Metabolism transforms nutrients into energy, repairs broken structures, and clears away waste. It is a dance of balance, flux, and renewal. But with time, this fire falters. The metabolic process is slow, the regulatory signals become confused, and the machinery begins to sputter. In this shifting landscape of energy and entropy, aging finds fertile ground. At the heart of this shift is the mitochondrion, a cellular organelle once thought to be nothing more than a power generator. In truth, it is far more, a conductor of cellular health, a sentinel of vitality, and a subtle architect of longevity. Mitochondria convert glucose and oxygen into adenosine triphosphate, or ATP, the molecular currency of energy. But the process is not perfect. It generates reactive oxygen species, molecules that, in excess, damage proteins, lipids, and DNA. When mitochondrial efficiency falters, the cell becomes polluted with waste and starved of energy. This mitochondrial dysfunction is a hallmark of aging. It leads to fatigue, muscle decline, neurodegeneration, and even cellular death. But the mitochondria are not passive victims. They can divide, fuse, and regenerate. They have their own genome, and they communicate with the nucleus in a two-way conversation of survival. When this conversation is corrupted, the result is metabolic chaos. Interventions that restore mitochondrial health have become a frontier of anti-aging science. Fasting and caloric restriction increase mitochondrial biogenesis, the creation of new mitochondria, and enhance their function. Compounds like nicotinamide riboside, NR, and nicotinamide mononucleotide, NMN, raise levels of NAD+, a coenzyme essential for mitochondrial activity and DNA repair. Exercise, especially endurance training, enhances mitochondrial density and efficiency. And emerging therapies aim to remove damaged mitochondria through a process called mitophagy, cleaning the cellular house from within. But mitochondria are not isolated players. They are deeply entwined with the endocrine system, the hormonal architecture of life. Aging disrupts this network in a phenomenon known as endocrine dysregulation. Growth hormone declines, insulin sensitivity wanes, cortisol patterns become erratic, and sex hormones diminish. This hormonal entropy leads to muscle wasting, fat accumulation, insulin resistance, sleep disturbances, and emotional volatility. Of particular interest is insulin, the master regulator of blood sugar. With age, tissues become less sensitive to insulin signals, leading to higher glucose levels and greater stress on the pancreas. This state, known as insulin resistance, is a precursor to metabolic syndrome, type 2 diabetes, and a cascade of degenerative processes. High glucose damages blood vessels, accelerates inflammation, and impairs cognition. Glycation, the binding of sugar to proteins, leads to the formation of advanced glycation end products, AGEs, which stiffen tissues, cloud lenses, and age the skin. Managing insulin resistance becomes central to longevity. Diets low in refined, carbohydrates rich in fiber and healthy fats, and attuned to glycemic response can reverse or delay this decline. Intermittent fasting reduces insulin levels and restores sensitivity. Physical activity, especially strength training, increases glucose uptake by muscle, reducing the burden on insulin. And novel pharmaceuticals, like metformin, modulate insulin action, showing promise not only in treating diabetes but in extending health span. Equally vital are the rhythms that govern these systems. The body is not a machine but a symphony, orchestrated by circadian clocks that regulate sleep, hormone release, metabolism, and immune function. These clocks are rooted in the suprachiasmatic nucleus of the brain, but every cell in the body contains its own circadian rhythm, synchronized to light, feeding, and activity. With age, these rhythms fade. Sleep becomes fragmented, hormone release becomes mistimed, and the synchronization between systems begins to unravel. This desynchronization accelerates aging, leading to metabolic disorder, cognitive decline, and even increased mortality. Restoring circadian alignment through light exposure, meal timing, and sleep hygiene becomes a tool of profound biological impact. Melatonin, the hormone of night, also declines with age. Known for regulating sleep, melatonin is also a powerful antioxidant and modulator of inflammation. Supplementation may restore not only rest, but also resilience against oxidative damage and age-related disease. Other clock-regulating compounds like chronobiotics aim to reset the circadian system, offering a pharmacological path to realignment. Yet metabolism is not merely a question of fuel and flame. It is also governed by nutrient sensing, the cell's ability to detect scarcity and abundance. Several pathways regulate this sensing: mTOR, AMPK, and sirtuins. These pathways do not merely react, they sculpt the cell's future. mTOR, or mechanistic target of rapamycin, promotes growth and reproduction in times of abundance. When inhibited, mTOR slows aging by enhancing autophagy, the recycling of damaged cellular components. Rapamycin, a drug that targets mTOR, has extended lifespan in animal models and is now being studied for its effects in humans. However, the balance is delicate, for excessive inhibition may impair wound healing and immune function. AMPK is the cell's energy sensor. Activated by low energy states such as fasting or exercise, AMPK enhances fat burning, mitochondrial activity, and glucose uptake. It counteracts the effects of overnutrition and promotes cellular renewal. Natural compounds like berberine and resveratrol, as well as pharmaceuticals like metformin, activate AMPK, mimicking the effects of caloric restriction. Sirtuins, a family of proteins dependent on NAD+, regulate gene expression, DNA repair, and inflammation. They are activated by fasting, cold exposure, and certain polyphenols. Sirtuin activity declines with age, but enhancing NAD+ levels can restore their function. The interplay between these pathways, mTOR, AMPK, sirtuins, is a molecular triangle at the heart of aging. Modulating them through behavior or biochemistry reshapes the trajectory of cellular time. Surrounding all of this is the extracellular matrix, the scaffolding that supports tissues and transmits signals between cells. With age, this matrix stiffens, calcifies, and loses its ability to transmit information. Fibrosis, a process of excessive, connective tissue buildup, impairs organ function and distorts architecture. The elasticity of youth gives way to the rigidity of age. Breaking this rigidity requires enzymatic intervention, mechanical stimulation, and sometimes cellular replacement. Antifibrotic therapies, including some derived from marine organisms and traditional medicine, are being explored for their ability to soften the matrix and restore tissue plasticity. The body, once thought to harden inevitably, may yet be coaxed into pliability. Even the fat that surrounds our organs shifts with time. Visceral fat, which increases with age, is metabolically active, not inert storage, but an endocrine organ in itself. It secretes inflammatory molecules, disrupts hormonal balance, and increases the risk of metabolic disease. Brown fat, by contrast, burns energy to generate heat and may protect against obesity and diabetes. Encouraging the browning of white fat through cold exposure, certain nutrients and pharmacology emerges as a new metabolic strategy. Thus, metabolism is no longer a static concept. It is dynamic, malleable, deeply tied to behavior, environment, and molecular nuance. It is not just about calories in and out, but about timing, quality, balance, and harmony. Aging, at its metabolic core, is a matter of rhythm disrupted, of fires fading, of signals crossed. But the orchestra can be retuned. In each of these interlocking systems, mitochondrial energy, insulin signaling, circadian regulation, nutrient sensing, and tissue integrity, we see a canvas of opportunity. Biology reveals not a closed fate, but a landscape of choices, levers to pull, clocks to reset. The metabolic flame need not dim in silence. It can be rekindled, guided, and even refined. To understand aging is not to fear it, but to navigate it, and in the understanding, to shape it. In every cell, a decision is made toward damage or repair, toward silence or renewal, toward decline or resilience. With knowledge comes agency, and with agency, a different kind of time begins. The human body is an orchestra of cellular communities, each playing its own tune yet harmonizing in a grand symphony. As time passes, however, the harmony frays and discord grows. Central to this decline is the immune system, our vigilant guardian, against infection, injury, and malignancy. Aging ushers in a state of immune remodeling known as immunosenescence, an intricate shift in immune function that paradoxically dampens defense while kindling chronic inflammation. This smoldering fire, within the body, sometimes called inflammaging, quietly accelerates tissue damage and underlies many age-related diseases. At the front line of immunity are white blood cells, lymphocytes, macrophages, neutrophils, each specialized to detect, neutralize, and remember threats. Yet with age, the diversity and responsiveness of lymphocytes decline. The thymus, a central organ for T-cell education, shrinks dramatically after adolescence, leading to fewer naive T-cells available to recognize new pathogens. Memory cells accumulate, but their functionality wanes, and the immune repertoire narrows. Simultaneously, innate immune cells like macrophages and neutrophils exhibit altered signaling and impaired clearance of cellular debris and pathogens. The communication between immune cells mediated by cytokines and chemokines becomes dysregulated. Instead of resolving inflammation, signals persist, creating a low-grade chronic inflammatory environment that harms tissues. This persistent inflammation contributes to the progression of cardiovascular disease, type 2 diabetes, neurodegeneration, and cancers. Markers like C-reactive protein and interleukin-6 rise steadily with age, correlating with frailty and mortality. The paradox lies in the coexistence of immune decline and hyperactivation, a system exhausted yet on high alert. Yet the immune system is adaptable. Lifestyle interventions, exercise, balanced diet, stress management, and adequate sleep can modulate inflammation and bolster immune competence. Physical activity stimulates the production of anti-inflammatory cytokines and improves circulation of immune cells. Diets rich in antioxidants, omega-3 fatty acids, and polyphenols help quench oxidative stress and reduce inflammatory markers. Emerging research into senolytics, drugs designed to selectively eliminate senescent cells, shows promise in dampening inflammaging. Senescent cells, despite their growth arrest, secrete a cocktail of pro-inflammatory factors known as the senescence-associated secretory phenotype, SASP, which perpetuates tissue dysfunction. Clearing these cells in animal models rejuvenates tissues and improves immune function. Vaccination strategies also adapt to the aging immune landscape. Higher dose or adjuvanted vaccines enhance immune responses in older adults, providing better protection against influenza, pneumonia, and shingles. Understanding the immune clock of aging will guide personalized medicine to strengthen defenses in the golden years. Beyond immune cells lies the bone marrow, the cradle of hematopoiesis, the birth of all blood cells. With age, hematopoietic stem cells decline in number and function, skewing differentiation toward myeloid cells and away from lymphoid lineage. This imbalance fuels inflammation and impairs adaptive immunity. Epigenetic changes, DNA damage, and altered niche signaling contribute to stem cell exhaustion. The lymphatic system, responsible for clearing fluids and waste from tissues, also experiences decline. Impaired lymphatic flow reduces immune surveillance and promotes tissue edema. The interplay between the immune and lymphatic systems is a crucial frontier in understanding aging and developing therapies. In the brain, microglia, the resident immune cells, shift from a resting to a pro-inflammatory state with age. This contributes to neuroinflammation, synaptic loss, and cognitive decline. modulating microglial activation through lifestyle, pharmaceuticals, or novel interventions offers hope for mitigating neurodegenerative diseases. Taken together, the aging immune system is not simply a failing defense, but a complex dynamic network undergoing profound remodeling. It is a balance between suppression and activation, exhaustion and hyperreactivity, vulnerability and resilience. Unlocking the secrets of immunosenescence reveals pathways not only to extend lifespan, but to enhance health span, the years lived free from chronic disease. Immunity and aging are intertwined in a delicate dance, shaped by genetics, environment, and choices. The body's internal sentinel can be nurtured and restored, its rhythms recalibrated, the storm of inflammation quelled, the defense line reinforced. In this lies a profound promise, not immortality, but a fuller, richer life in the years we are granted. Beneath the visible decline of muscles and bones lies a silent transformation that shapes our physical form and strength over time. The musculoskeletal system, once a pillar of vitality and mobility, gradually succumbs to the wear of years. Sarcopenia, the age-related loss of muscle mass and function, is not merely a cosmetic change. It is a fundamental shift that influences balance, metabolism, and independence. Muscles are intricate tissues composed of fibers, connective tissue, nerves, and blood vessels, all working in concert to generate force and movement. As aging progresses, muscle fibers diminish in size and number, especially the fast twitch fibers responsible for quick, powerful actions. This loss is accompanied by infiltration of fat and connective tissue, which further impairs function. The causes of sarcopenia are multifaceted. Declining anabolic hormones, such as testosterone, growth hormone, and insulin-like growth factor 1 reduce muscle protein synthesis. Neural factors, including reduced motor neuron number and altered neuromuscular junctions, weaken the communication between nerves and muscle fibers. Inflammatory cytokines, oxidative stress, and mitochondrial dysfunction compound the decline, creating an environment hostile to muscle regeneration. Physical inactivity accelerates muscle wasting, creating a vicious cycle where weakness leads to less movement, further degradation, and increased risk of falls and fractures. Conversely, resistance training stands as a powerful intervention, stimulating muscle hypertrophy, enhancing neural adaptation, and improving metabolic health. Even in advanced age, muscles respond to exercise, underscoring the remarkable plasticity of the human body. Nutrition also plays a pivotal role. Adequate protein intake, particularly essential amino acids like leucine, stimulates muscle protein synthesis. Vitamin D, omega-3 fatty acids, and antioxidants support muscle health by modulating inflammation and promoting repair. Emerging evidence suggests that timing of protein intake around exercise may maximize muscle gains. Bone, the scaffold of the body, undergoes its own transformation. Peak bone mass is reached by early adulthood, after which resorption gradually outpaces formation. Osteoporosis, the reduction in bone density and quality, increases fragility and fracture risk. The interplay between osteoblasts, bone builders, and osteoclasts, bone resorbers, becomes imbalanced, influenced by hormonal changes, inflammation, and lifestyle. Calcium and vitamin D are foundational to bone health, facilitating mineralization and remodeling. Weight-bearing exercise applies mechanical stress that stimulates bone formation, counteracting the tendency toward loss. Medications like bisphosphonates and selective estrogen receptor modulators have been developed to treat osteoporosis, yet prevention through lifestyle remains paramount. Joints, the points of articulation, often suffer from osteoarthritis, a degenerative condition marked by cartilage breakdown, inflammation, and pain. Cartilage, devoid of blood vessels and nerves, relies on diffusion and joint movement for nutrition. Aging impairs cartilage repair, alters synovial fluid composition, and promotes low-grade inflammation, leading to stiffness and disability. Maintaining joint health involves regular, moderate exercise, weight management, and anti-inflammatory nutrition. Advances in regenerative medicine, such as stem cell therapy and tissue engineering, seek to restore cartilage and joint function, offering hope beyond symptom management. The musculoskeletal system is not isolated. It communicates with other systems through mechanical signals, hormones, and inflammatory mediators. Muscle-derived myokines influence metabolism and immunity, while bone releases osteokines affecting energy balance and cognition. This intricate crosstalk emphasizes the body's unity and the importance of preserving musculoskeletal integrity. As the body ages, maintaining strength and mobility becomes essential not only for physical function, but also for psychological well-being and social engagement. The loss of independence wrought by musculoskeletal decline impacts quality of life profoundly. Yet within this decline lies resilience, the capacity to rebuild muscle, strengthen bones, and soothe joints persists. Through movement, nourishment, and care, the body invites renewal. Sarcopenia and osteoporosis are not inevitabilities, but challenges met with knowledge and action. In this lifelong journey, the musculoskeletal system stands as a testament to the body's ability to adapt and endure. Its preservation is a cornerstone of healthy aging, a foundation upon which the fullness of life can be built, step by careful step. As the body ages, the vital dance of metabolism slows, and the cell's capacity to convert nutrients into energy diminishes, reshaping how we live and age. Metabolism, the sum of all chemical reactions sustaining life, is the engine that powers growth, repair, and movement. At its heart lie the mitochondria, the tiny organelles within each cell often called the powerhouses of life. These microscopic dynamos convert oxygen and nutrients into adenosine triphosphate, ATP, the energy currency that fuels every biological process. With age, mitochondrial function declines in both quantity and quality. Mitochondria become fewer, smaller, and less efficient. Their membranes lose integrity and their DNA accumulates mutations. This mitochondrial dysfunction leads to reduced energy production, increased generation of reactive oxygen species, ROS, and impaired regulation of cell death and inflammation. The excess ROS, highly reactive molecules derived from oxygen, can damage proteins, lipids, and DNA, contributing to oxidative stress. While low levels of ROS serve important signaling roles, chronic oxidative stress disrupts cellular homeostasis and accelerates aging. Mitochondrial DNA, more vulnerable than nuclear DNA due to its proximity to ROS and limited repair mechanisms, accumulates mutations over time, undermining mitochondrial integrity further. This self-perpetuating cycle of mitochondrial damage and oxidative stress plays a central role in aging and age-related diseases including neurodegeneration, cardiovascular disease, diabetes, and sarcopenia. The decline in mitochondrial function also impairs metabolic flexibility, reducing the ability to switch efficiently between carbohydrates and fats as fuel sources, which affects overall energy balance and body composition. Yet the story of mitochondria and aging is not solely one of decline. The cell possesses mechanisms of mitochondrial quality control, biogenesis, fusion, fission, and mitophagy that maintain a healthy population of mitochondria. Biogenesis, stimulated by factors such as exercise and caloric restriction, increases the number of mitochondria. Fusion and fission allow damaged mitochondria to merge with healthy ones, or split to isolate defective parts. Mitophagy selectively removes dysfunctional mitochondria, preventing accumulation of damaged organelles. Interventions that activate these quality control processes show promise for promoting healthy aging. Physical exercise, especially endurance training, robustly stimulates mitochondrial biogenesis and improves mitochondrial efficiency. Caloric restriction and intermittent fasting activate cellular stress responses that enhance mitochondrial turnover and function. Certain compounds like resveratrol, nicotinamide riboside, and metformin are under investigation for their potential to boost mitochondrial health and extend lifespan. The metabolic rate, the speed at which the body burns calories, also changes with age. Resting metabolic rate tends to decline, influenced by loss of lean body mass, hormonal shifts, and reduced mitochondrial function. This decline can contribute to weight gain and metabolic disorders if caloric intake remains unchanged. Adjusting diet and activity levels becomes essential to maintain metabolic balance. Cellular metabolism extends beyond energy production to include nutrient-sensing pathways, molecular circuits that detect and respond to availability of glucose, amino acids, and fats, key regulators like mTOR, mechanistic target of rapamycin, AMPK, AMP-activated protein kinase, and sirtuins integrate signals to coordinate growth, autophagy, and stress responses. Dysregulation of these pathways is implicated in aging and metabolic diseases. Caloric restriction, known to extend lifespan in multiple species, modulates these nutrient sensors by limiting energy intake without malnutrition, triggering protective cellular responses. Pharmaceutical mimetics of caloric restriction aim to harness these effects, potentially promoting metabolic health and longevity. The intricate interplay between metabolism, mitochondria, and aging reveals the profound importance of cellular energy balance. Each breath, each beat, each movement depends on the delicate orchestration of these processes. The decline with age is not merely a fading light, but a call to understand and intervene. By nurturing mitochondrial health through lifestyle and emerging therapies, we may unlock new possibilities, vitality sustained, disease delayed, life extended. The energy that powers our existence need not wane in the twilight years, but can be rekindled to illuminate a path toward a fuller, healthier life. The human brain, a marvel of complexity and subtlety, is not immune to the passage of time. Aging weaves its threads through neural circuits, shaping cognition, memory, and emotion. The biology of brain aging is a tapestry of changes, some gradual and benign, others insidious and profound, that together influence how we think, feel, and connect. At the cellular level, neurons, the primary signaling units of the brain, show altered structure and function. Dendrites, the branched extensions receiving signals, retract and lose spines, reducing synaptic connections. Synapses themselves, the specialized junctions for communication, diminish in number and efficacy. This synaptic loss impairs neural plasticity, the brain's capacity to adapt and reorganize in response to experience. Neurotransmitter systems change with age. Levels of dopamine, acetylcholine, serotonin, and norepinephrine fluctuate, affecting mood, attention, motivation, and memory. The reduction of dopamine in particular is linked to slowed movement and cognitive processing, contributing to conditions like Parkinson's disease and age-associated cognitive decline. White matter, the myelinated fibers that connect brain regions, undergoes degradation, disrupting communication networks. Myelin loss, axonal damage, and microvascular changes impair signal transmission, reflected in slower processing speed and reduced coordination. The blood-brain barrier, a protective interface regulating substances entering the brain, becomes more permeable, increasing vulnerability to toxins and inflammation. Mitochondrial dysfunction and oxidative stress accumulate in brain cells, contributing to neuronal damage and death. Protein homeostasis, the delicate balance of synthesis, folding, and clearance falters, leading to accumulation of misfolded proteins such as beta-amyloid and tau. These aggregates are hallmarks of Alzheimer's disease, the most common form of dementia. Inflammation within the brain, mediated by activated microglia and astrocytes, shifts from protective to chronic and damaging. This neuroinflammation exacerbates neuronal injury and interferes with repair processes. The interplay between immune aging and brain aging is a vital area of study, revealing how systemic inflammation influences cognition. Despite these challenges, the brain maintains remarkable plasticity throughout life. Neurogenesis, the birth of new neurons, persists in regions like the hippocampus, essential for learning and memory. Cognitive reserve, built through education, mental stimulation, and social engagement, buffers against decline and delays onset of clinical symptoms. Lifestyle factors powerfully influence brain aging. Regular physical exercise enhances blood flow, promotes neurogenesis, and upregulates neurotrophic factors such as BDNF, brain-derived neurotrophic factor. A diet rich in antioxidants, omega-3 fatty acids, and polyphenols supports neuronal health and reduces inflammation. Social interaction and lifelong learning stimulate neural networks and build cognitive resilience. Sleep, often overlooked, plays a critical role in brain maintenance. During deep sleep stages, the brain's glymphatic system clears metabolic waste, including beta-amyloid. Disrupted sleep accelerates cognitive decline and increases risk of neurodegenerative diseases. Advances in neuroscience reveal exciting therapeutic avenues. Pharmacological agents targeting amyloid and tau, anti-inflammatory drugs, and lifestyle interventions show promise in mitigating age-related cognitive decline. Brain stimulation techniques and cognitive training are being explored to enhance neural function. Understanding brain aging is not merely an academic pursuit, but a profound human imperative. The essence of identity, memory, creativity, and connection lies within this organ. To preserve brain health is to preserve the very self. The biological story of brain aging is one of vulnerability and resilience, loss and adaptation. By embracing knowledge and proactive care, we nurture the mind's capacity to thrive across decades. The mind's voyage through time can remain rich, vibrant, and meaningful, a testament to the enduring spirit of human life. The immune system, our vigilant guardian against disease and injury, undergoes profound changes as the years advance, shaping the aging process in ways both visible and subtle. This transformation, known as immunosenescence, reflects a gradual decline in immune competence accompanied paradoxically by chronic low-grade inflammation, a state termed inflammaging. Together, these shifts influence susceptibility to infections, cancer, autoimmune conditions, and the overall trajectory of aging. Immunosenescence affects both the innate and adaptive arms of immunity. The innate immune system, the first line of defense, relies on cells such as macrophages, neutrophils, dendritic cells, and natural killer cells to rapidly recognize and eliminate threats. With age, their ability to respond swiftly diminishes. Phagocytosis, the engulfing of pathogens, is less efficient, and the production of signaling molecules like cytokines becomes dysregulated. The adaptive immune system, responsible for specific, long-lasting protection, also falters. T lymphocytes, which coordinate immune responses and directly attack infected or malignant cells, decline in number and function. The thymus, the organ where T cells mature, shrinks dramatically after puberty, reducing the output of new naive T cells. This shrinkage limits the body's capacity to recognize novel pathogens, contributing to poorer vaccine responses and increased infection risk. B lymphocytes, producers of antibodies, exhibit altered diversity and responsiveness. The repertoire of antibodies narrows, reducing the ability to combat new antigens effectively. Memory B cells may persist, but their function is compromised, impairing long-term immunity. The dual phenomena of immunosenescence and inflammaging are intertwined. Chronic activation of the immune system leads to persistent, low-level inflammation, marked by elevated levels of inflammatory cytokines, such as IL-6, TNF-alpha, and CRP. This inflammation contributes to tissue damage, promotes age-related diseases, including atherosclerosis, diabetes, and neurodegeneration, and interferes with repair mechanisms. Sources of inflammaging include the accumulation of senescent cells that secrete pro-inflammatory factors, chronic infections such as cytomegalovirus, and changes in the gut microbiome that affect systemic immunity. The microbiota, the community of microorganisms residing primarily in the gut, shifts in composition and diversity with age, influencing inflammation and immune regulation. Lifestyle factors exert a powerful influence on immune aging. Nutrition, rich in antioxidants, vitamins, notably vitamins C, D, and E, and minerals, supports immune function. Regular moderate exercise enhances immune surveillance and reduces inflammation, while chronic stress and poor sleep undermine immune resilience. Emerging research explores interventions to modulate immunosenescence and inflammaging. Senolytics, drugs that selectively eliminate senescent cells, show promise in reducing inflammatory burden and restoring tissue function. Vaccination strategies are being optimized for the elderly, incorporating adjuvants to boost immune responses. Probiotics and dietary fibers aim to restore microbiome balance and improve immune health. Understanding the immune system's transformation with age reveals both vulnerability and opportunity. It underscores the importance of preventive care, timely vaccinations, and lifestyle optimization in promoting healthy aging. The immune system's dance between defense and inflammation shapes the body's internal environment. In the balance lies the potential to resist disease, repair damage, and maintain vitality. To age well is to nurture this balance, to honor the body's guardian, and guide it with wisdom, compassion, and care. Aging is not solely the tale of decline. It is also a story of cellular endurance and transformation. At the heart of this narrative lies cellular senescence, a state where cells cease to divide and adopt a distinctive phenotype that profoundly influences tissue function and systemic aging. Senescent cells, once thought to be merely inert bystanders, have emerged as key players in the biology of aging and age-related diseases. Cellular senescence is triggered by various stressors: DNA damage, telomere shortening, oxidative stress, oncogene activation, and mitochondrial dysfunction that signal a cell to exit the cell cycle permanently. This process acts as a tumor-suppressive mechanism, preventing damaged cells from proliferating and potentially becoming cancerous. However, senescent cells do not simply vanish. They linger, altering their microenvironment through the senescence-associated secretory phenotype, SASP. SASP is characterized by the secretion of pro-inflammatory cytokines, chemokines, growth factors, and proteases. While initially beneficial in wound healing and immune surveillance, persistent SASP fosters chronic inflammation, tissue remodeling, and degradation of extracellular matrix. The accumulation of senescent cells contributes to functional decline in organs, impaired regeneration, and promotes a milieu conducive to cancer and degenerative diseases. The burden of senescent cells increases with age, particularly in tissues with limited regenerative capacity, such as the skin, lungs, vasculature, and adipose tissue. This accumulation disrupts tissue homeostasis and impairs the stem cell niche, reducing the body's ability to repair damage and respond to stress. Emerging therapeutic strategies seek to target senescent cells to mitigate their detrimental effects. Senolytics, a novel class of drugs, selectively induce apoptosis in senescent cells, thereby reducing SASP and improving tissue function in animal models. Early clinical trials show promise in conditions like idiopathic pulmonary fibrosis and osteoarthritis. Beyond pharmacological approaches, lifestyle factors influence senescence. Caloric restriction, physical exercise, and certain dietary components may reduce the accumulation or impact of senescent cells by decreasing oxidative stress and inflammation. Senescence also intersects with epigenetic alterations, the changes in gene expression regulation that occur without altering the DNA sequence. Aging cells exhibit shifts in DNA methylation, histone modification, and chromatin remodeling, affecting the activation of senescence pathways and the SASP profile. Understanding the dual role of cellular senescence as protector and promoter of aging reveals a complex balance. It is a defense mechanism turned double-edged sword, where the failure to clear senescent cells leads to a pro-aging environment. The biology of cellular senescence invites a reimagining of aging as a dynamic process, where intervention can tip the scales toward health and longevity. By targeting the cells that hold the keys to aging's secrets, science opens new avenues for extending the health span, transforming how we age and live. As we delve deeper into the biology of aging, the role of stem cells emerges as a cornerstone of tissue maintenance and regeneration. Stem cells possess the remarkable ability to self-renew and differentiate into various specialized cell types, sustaining organ function throughout life. Yet with age, the number, function, and regenerative capacity of stem cells decline, contributing to the gradual loss of tissue integrity and repair. Adult stem cells reside in specialized niches, microenvironments that regulate their behavior and protect them from stress. Hematopoietic stem cells in the bone marrow replenish blood and immune cells. Mesenchymal stem cells contribute to bone, cartilage, and fat. Neural stem cells in the brain support neurogenesis. These cells are essential for replacing damaged or aged cells, maintaining homeostasis, and adapting to physiological demands. Aging stem cells encounter numerous challenges. DNA damage accumulates, telomeres shorten, and epigenetic alterations modify gene expression patterns, leading to decreased proliferation and differentiation potential. The stem cell niche itself undergoes remodeling, often becoming less supportive, due to changes in extracellular matrix composition, vascularization, and signaling molecules. Senescence and inflammation also impair stem cell function. Exposure to SASP factors from neighboring senescent cells can induce stem cell exhaustion or aberrant differentiation, disrupting tissue regeneration. Chronic inflammation, characteristic of aging, skews stem cell fate decisions, favoring fibrotic or adipogenic outcomes over functional tissue repair. Mitochondrial dysfunction and metabolic shifts affect stem cell energetics, influencing their capacity to divide and differentiate. Druid stem cells often exhibit altered energy metabolism, favoring glycolysis over oxidative phosphorylation, which may impact their functionality and longevity. Despite these challenges, the stem cell compartment retains some plasticity. Certain interventions can rejuvenate aged stem cells or enhance their function. Caloric restriction and exercise stimulate stem cell activity and improve niche health. Pharmacological agents targeting signaling pathways like WNT, Notch, and mTOR show potential in restoring regenerative capacity. Cellular reprogramming technologies, such as induced pluripotent stem cells, iPSCs, offer revolutionary possibilities for regenerating tissues and studying aging mechanisms. However, safely translating these advances into therapies remains a formidable challenge. The decline in stem cell function with age underlies many age-associated pathologies: anemia, osteoporosis, sarcopenia, neurodegeneration, and impaired wound healing. Understanding the molecular and environmental drivers of stem cell aging is critical to developing strategies that maintain tissue health and extend the regenerative potential throughout life. Stem cells, the architects of renewal, hold the promise of reversing or slowing age-related decline. By nurturing these cellular fountains of youth, we can aspire to prolong vitality, resilience, and function, the essence of healthy aging. Mitochondria, often called the powerhouses of the cell, are central to the biology of aging. These tiny organelles generate the energy currency, adenosine triphosphate, ATP, fueling cellular processes vital for survival and function. Yet mitochondria are more than mere energy factories. They regulate metabolism, apoptosis, calcium signaling, and the production of reactive oxygen species, ROS. As we age, mitochondrial function declines, contributing profoundly to cellular senescence, tissue dysfunction, and the aging phenotype. Mitochondrial DNA, mtDNA, is distinct from nuclear DNA, encoding essential components of the respiratory chain. Unlike nuclear DNA, mtDNA is more vulnerable to damage because of its proximity to the electron transport chain where ROS are generated and its limited repair mechanisms. Accumulation of mutations and deletions in mtDNA impairs mitochondrial function, leading to decreased ATP production and increased oxidative stress. Oxidative stress arises when the generation of ROS exceeds the cell's antioxidant defenses. ROS, while vital signaling molecules at physiological levels, become damaging when in excess, oxidizing proteins, lipids, and DNA. This oxidative damage compromises cellular structures and activates stress response pathways, promoting inflammation and senescence. Mitochondrial dynamics, the balance of fusion and fission processes, maintain mitochondrial integrity and function. Aging disrupts these dynamics, leading to fragmented or enlarged mitochondria that are less efficient. Mitophagy, the selective autophagic removal of damaged mitochondria, also declines with age, resulting in accumulation of dysfunctional mitochondria. Energy metabolism shifts with age, often resulting in reduced metabolic flexibility. Cells become less efficient at switching between fuel sources, impacting overall organismal energy balance. These metabolic changes contribute to age-associated diseases, including metabolic syndrome, neurodegeneration, and cardiovascular disease. Mitochondrial dysfunction also intersects with cellular signaling pathways that regulate aging, such as AMP-activated protein kinase, AMPK, mammalian target of rapamycin, MTR, and sirtuins. Modulating these pathways has been shown to improve mitochondrial function and extend lifespan in model organisms. Lifestyle factors play a critical role in mitochondrial health. Regular exercise stimulates mitochondrial biogenesis, the generation of new mitochondria, and enhances antioxidant capacity. Dietary interventions like caloric restriction or intermittent fasting improve mitochondrial efficiency and reduce oxidative damage. Pharmacological agents targeting mitochondria are under intense investigation. Compounds such as NAD+ precursors, antioxidants, and mitochondrial-targeted peptides show potential in restoring mitochondrial function and ameliorating age-related decline. Mitochondria stand at the crossroads of life and aging, their vitality reflecting cellular and organismal health. By preserving mitochondrial function, we can influence the fundamental processes of aging, promoting longevity and wellness. Telomeres, the protective caps at the ends of chromosomes, play a pivotal role in the biology of aging by safeguarding genomic integrity during cell division. These repetitive DNA sequences, coupled with specialized proteins, prevent chromosomes from fraying or fusing with one another, much like the plastic tips of shoelaces prevent unraveling. Yet each time a cell divides, telomeres shorten slightly, serving as a biological clock that limits the number of times a cell can replicate, a phenomenon known as the Hayflick limit. Telomere shortening acts as a double-edged sword. On one hand, it prevents uncontrolled cell proliferation, acting as a tumor suppressor mechanism by triggering cellular senescence or apoptosis when telomeres become critically short. On the other hand, excessive shortening leads to impaired tissue regeneration and contributes to the functional decline associated with aging. Several factors accelerate telomere attrition beyond the natural cell division process. Chronic stress, inflammation, oxidative damage, and unhealthy lifestyle choices such as smoking, poor diet, and physical inactivity exacerbate telomere shortening. Conversely, interventions like regular exercise, stress reduction, and diets rich in antioxidants may slow this process. The enzyme telomerase can elongate telomeres, counteracting their shortening. While highly active in embryonic stem cells and certain immune cells, telomerase activity is limited in most somatic cells, a trade-off that balances cancer risk with tissue maintenance. Reactivating telomerase holds therapeutic promise but raises concerns about potential oncogenic effects. Shortened telomeres have been linked to a spectrum of age-related diseases, including cardiovascular disease, diabetes, osteoporosis, and neurodegeneration. Telomere length in leukocytes is often used as a biomarker of biological aging and disease risk, though the relationship is complex and influenced by genetic and environmental factors. Beyond length, telomere dysfunction involves structural changes, and altered interactions with shelterin proteins that protect chromosome ends. Damage to telomeric DNA can trigger DNA damage responses, promoting senescence and inflammation. Recent research explores novel approaches to protect and restore telomere function, including gene therapy, small molecules that activate telomerase, and lifestyle modifications that reduce oxidative stress. The intricate balance between telomere maintenance, cellular aging, and cancer suppression continues to challenge scientists. Telomeres, the ticking clocks of cellular life, mark the passage of time at the molecular level. Their gradual erosion encapsulates the essence of aging, a high balance between renewal and limitation. Understanding and influencing telomere dynamics offers a window into extending healthy lifespan while preserving the delicate harmony of life. The intricate dance of epigenetics, the chemical modifications that regulate gene expression without altering the underlying DNA sequence, emerges as a fundamental mechanism shaping the aging process. Epigenetic changes orchestrate which genes are turned on or off, influencing cellular identity, function, and adaptation. As we age, these regulatory patterns shift, contributing to cellular dysfunction, loss of tissue homeostasis, and age-related diseases. Key epigenetic modifications include DNA methylation, histone modifications, and chromatin remodeling. DNA methylation involves the addition of methyl groups to cytosine bases in DNA, often silencing gene expression. With age, global hypomethylation occurs alongside site-specific hypermethylation, disrupting normal gene regulation. These changes can activate harmful genes or silence protective ones, impairing cellular function. Histones, the protein spools, around which DNA wins, undergo post-translational modifications such as acetylation, methylation, and phosphorylation. These modifications alter chromatin structure, influencing gene accessibility. Age-associated shifts in histone marks can loosen or tighten chromatin, affecting genomic stability and transcriptional programs. Chromatin remodeling complexes reposition nucleosomes, dynamically regulating access to DNA. Aging disrupts the balance of these complexes, further contributing to epigenetic drift. This drift, an accumulation of stochastic epigenetic alterations, leads to increased cellular heterogeneity and loss of youthful gene expression patterns. Epigenetic changes not only reflect aging but can drive it. Aberrant methylation and histone modifications influence stem cell function, immune responses, and cellular senescence. They also intersect with other aging hallmarks, including telomere attrition, mitochondrial dysfunction, and DNA damage. Remarkably, the epigenome retains a degree of plasticity. Environmental factors, lifestyle, and pharmacological agents can modulate epigenetic marks. Nutrients such as folate, B vitamins, and polyphenols participate in methylation processes, while compounds like histone deacetylase inhibitors show potential in rejuvenating aged cells. The concept of an epigenetic clock, based on methylation patterns, provides a powerful biomarker for biological age, predicting morbidity and mortality more accurately than chronological age. This tool underscores the centrality of epigenetics in aging research and offers a framework for testing interventions. Reprogramming aged cells to a more youthful epigenetic state, without erasing their identity, represents a frontier in regenerative medicine. Partial reprogramming has been shown to reverse some aging features in cells and tissues, heralding new possibilities for extending health span. Epigenetics bridges the genome and environment, encoding life's experiences in chemical marks. Its dynamic nature offers hope that aging is not an immutable fate but a malleable journey, shaped by choices and innovations. The immune system, our guardian against pathogens and internal threats, undergoes profound transformations with age, a process known as immunosenescence. This decline in immune function not only increases susceptibility to infections, but also fuels chronic inflammation, termed inflammaging, which contributes to many age-related diseases. Immunosenescence involves both the innate and adaptive arms of immunity. The innate immune system, the body's first line of defense, shows altered function with age. Macrophages, dendritic cells, and natural killer cells exhibit reduced responsiveness and impaired pathogen clearance. Their ability to produce signaling molecules that orchestrate immune responses diminishes, weakening early defense mechanisms. The adaptive immune system, which provides targeted and long-lasting protection, experiences marked decline. T cells, particularly naive T cells that respond to new infections, decrease in number due to thymic involution, the gradual shrinking of the thymus gland with age. This reduction limits the body's ability to recognize and combat novel pathogens. Memory T cells, which respond to previously encountered antigens, accumulate but may become dysfunctional. B cells, responsible for producing antibodies, also decline in function. Their ability to generate high-affinity antibodies wanes, reducing vaccine efficacy and increasing vulnerability to infections. Alongside diminished defense, chronic low-grade inflammation persists in older adults. This inflammaging results from multiple sources: accumulation of senescent cells secreting pro-inflammatory factors, persistent infections, altered gut microbiota, and age-related changes in metabolism. While inflammation is vital for defense and repair, its chronic presence damages tissues and promotes diseases such as atherosclerosis, Alzheimer's, diabetes, and cancer. Regulatory mechanisms that normally restrain inflammation weaken with age, exacerbating the problem. The balance between pro- and anti-inflammatory signals shifts unfavorably, leading to a systemic pro-inflammatory state. Interventions targeting immunosenescence and inflammaging are being explored. Caloric restriction, exercise, and certain nutraceuticals show promise in modulating immune function. Drugs like rapamycin and metformin may improve immune responses by influencing underlying molecular pathways. Vaccination strategies tailored to older adults, including higher antigen doses or adjuvants, improve protection. Additionally, therapies aiming to clear senescent cells or modulate the microbiome could reduce inflammaging and enhance immunity. The aging immune system illustrates a paradox: diminished ability to defend coupled with persistent, harmful inflammation. Deciphering and mitigating these changes are vital to extending healthy lifespan and improving quality of life. Cellular. Senescence is a defining hallmark of aging, representing a state of permanent cell cycle arrest that cells enter in response to various stressors. Unlike apoptotic cell death, senescent cells remain metabolically active but no longer divide. While initially protective, preventing damaged cells from proliferating and contributing to cancer, senescence becomes detrimental when senescent cells accumulate in tissues over time. Senescent cells secrete a complex mix of pro-inflammatory cytokines, chemokines, growth factors, and proteases, collectively known as the senescence-associated secretory phenotype, or SASP. This secretory profile disrupts tissue microenvironments, promotes inflammation, and can induce senescence in neighboring cells, creating a vicious cycle that accelerates tissue aging and dysfunction. The triggers of senescence are diverse: DNA damage, oxidative stress, telomere shortening, oncogene activation, and mitochondrial dysfunction all contribute. Senescent cells are found in nearly all age tissues and are implicated in various age-related diseases including osteoarthritis, atherosclerosis, pulmonary fibrosis, and neurodegeneration. The body's immune system normally clears senescent cells, but with age, immune surveillance declines, allowing these cells to accumulate. Their presence impairs tissue regeneration, alters stem cell niches, and compromises organ function. Recent breakthroughs in aging research focus on senolytics, drugs designed to selectively eliminate senescent cells, and cynomorphics, agents that suppress the harmful SASP without killing the cells. In animal models, senolytics have improved health span and reduced signs of age-related diseases, sparking hope for translation to humans. Moreover, lifestyle interventions such as exercise and caloric restriction may modulate senescence and SASP production. Understanding the complex interplay between senescence, immune function, and tissue homeostasis is essential for developing effective therapies. Cellular senescence, once viewed solely as a tumor suppressor mechanism, is now recognized as a double-edged sword, essential for protection but capable of driving aging and disease when unchecked. Targeting senescence represents a promising avenue to rejuvenate tissues and promote healthy aging. Proteostasis, the maintenance of cellular protein homeostasis, is fundamental to healthy cell function and longevity. Proteins, the workhorses of the cell, must fold into precise three-dimensional structures to perform their roles. Misfolded or damaged proteins, if not properly managed, accumulate and aggregate, disrupting cellular processes and contributing to aging and disease. Cells employ intricate quality control systems to preserve proteostasis. Molecular chaperones assist in protein folding, refolding, or targeting damaged proteins for degradation. The ubiquitin proteasome system and autophagy pathways selectively degrade misfolded proteins and damaged organelles, preventing toxic buildup. With aging, these proteostasis networks decline in efficiency. Chaperone expression decreases, proteasome activity wanes, and autophagic capacity diminishes. This leads to the accumulation of protein aggregates, such as amyloid plaques and neurofibrillary tangles observed in neurodegenerative diseases like Alzheimer's and Parkinson's. Proteostasis. Failure affects multiple tissues and contributes to functional decline. Protein aggregates interfere with cellular signaling, mitochondrial function, and membrane integrity. They trigger stress responses that can lead to apoptosis or senescence. Interventions aiming to restore proteostasis are under investigation. Enhancing autophagy through caloric restriction, exercise, or pharmacological agents like rapamycin shows promise in clearing damaged proteins. Molecular chaperones and proteasome activators are also potential therapeutic targets. The interplay between proteostasis and other aging hallmarks, such as mitochondrial dysfunction and cellular senescence, highlights the complexity of aging biology. Maintaining protein quality control is crucial for cellular health, resilience, and longevity. Proteostasis, the delicate balance of protein synthesis, folding, and degradation, serves as a guardian of cellular integrity. Preserving this balance offers a pathway to mitigate age-related decline and sustain vitality across the lifespan. At the nexus of aging research lies the concept of resilience, the ability of cells, tissues, and organisms to adapt, recover, and maintain function in the face of stress. Aging reflects a gradual erosion of this resilience, where accumulated damage and declining repair mechanisms tip the balance toward dysfunction and disease. Yet, the human body exhibits remarkable plasticity, offering hope that aging is not an unalterable fate but a modifiable process. Key molecular pathways regulate this resilience, including nutrient-sensing networks like insulin IGF-1 signaling, mTOR, AMPK, and sirtuins. These pathways integrate environmental cues, energy status, and stress signals to orchestrate metabolism, growth, and repair. Modulating their activity through lifestyle interventions or therapeutics can enhance longevity and health span. Hormesis, the biological phenomenon where low levels of stress stimulate adaptive beneficial responses, exemplifies resilience. Mild stressors such as exercise, caloric restriction, temperature fluctuations, and phytochemicals activate protective pathways, boosting cellular defenses, repair mechanisms, and metabolic efficiency. The emerging field of regenerative medicine aims to restore resilience by repairing or replacing damaged tissues using stem cells, gene therapy, and bioengineering. Advances in understanding stem cell aging and rejuvenation hold promise for combating age-related tissue decline. Epigenetic reprogramming offers another frontier, potentially resetting aged cells to a more youthful state without losing their identity, thereby restoring function. Senolytics and interventions targeting proteostasis and mitochondrial function further contribute to enhancing resilience. Ultimately, aging is a complex interplay of damage accumulation and repair, decline and adaptation. While the inevitability of aging remains, its pace and impact can be modulated. By embracing strategies that bolster resilience through science, lifestyle, and innovation, we unlock the potential for longer, healthier lives. The biology of aging reveals a profound truth. Within the cells and molecules that compose us lies not only the story of decline, but the blueprint for renewal. Through understanding and intervention, we stand at the threshold of redefining what it means to grow old.